Anatomy 32, Chapter 21, Special Senses, Topics, Hearing. So, the structures for hearing are housed in the temporal bones, or connected sort of directly to the temporal bone. So, the anatomy of the ear begins with the external ear. The external ear includes the auricle, the fleshy part of the ear on the outside that we can get pierced, including the helix, where the elastic cartilage is, and the lobule, which is mostly adipose tissue. And the auricle acts as a funnel to funnel sound waves into a canal. That canal is called the external auditory canal, and it'll carry the sound waves down the canal deeper into the head until the sound waves hit a structure called the tympanic membrane. And the tympanic membrane is a thin, semi-transparent partition, and it vibrates when hit by sound waves. That then brings us to the middle ear. The middle ear is an air-filled cavity that connects to the pharynx via the auditory tube. Within the middle ear are three auditory ossicles, these three tiny bones that will transmit the vibration from the tympanic membrane to the inner ear. This includes the malleus, which is directly attached to the tympanic membrane, then the incus, and then the stapes, which then fits into the oval window leading to the inner ear. So when the tympanic membrane vibrates, it causes the malleus to vibrate, which causes the incus to vibrate, which causes the stapes to vibrate. There is a oval window that the stapes fits into that leads to the inner ear, and that is a membrane structure that allows the vibration to pass through. There's also a round window that allows, that's a membrane structure that allows excess vibrations a way to escape the inner ear. And then, as I mentioned already, the auditory tube that connects the middle ear to the nasopharynx. And the auditory tube is important because it helps equalize air pressure on both sides of the tympanic membrane. Normally this is not noticeable at all, but if you are in an airplane and you feel your ears popping, that's when the auditory tube is equalizing that pressure. Then we have the internal ear. The internal ear is a series of fluid-filled canals. So when we look at the inner ear or internal ear, we see that it is a bony structure, and the bony structure is called the bony labyrinth, and that bony structure is filled with a fluid called the perilymph. And the bony labyrinth can be broken up into three areas: the semicircular canals, the vestibule, and the cochlea. The semicircular canals contain structures called semicircular ducts within the semicircular canals, and these are membrane structures. The vestibule houses the oval window, and the cochlea is a bony spiral that has a spiraling membrane within it, and also opens up via the round window. Now, within the bony labyrinth, as I've sort of mentioned already, is this membranous labyrinth made up of various membrane structures that are partially or completely connected to each other, and there's fluid inside of the membranous labyrinth, and that's called endolymph. So endolymph inside the membranous labyrinth, perilymph outside of the membranous labyrinth. And when we look at these circular canals, we see that they have these little widened areas called the semicircular ducts ampulla, so the ampulla, and that is where we find the structures that detect dynamic equilibrium. Within the vestibule, we have two membrane structures called the utricle and the saccule. The utricle and the saccule houses the structures that detect static equilibrium. And then, as we mentioned already, in the cochlea is the cochlear duct, a membrane structure that houses the structures for detecting hearing. Here is our model helix, the lobule, forming parts of the auricle, the external auditory canal leading to the tympanic membrane. Here we have the malleus attached to the tympanic membrane. Here are the inner ear with the semicircular canals, the vestibule, the cochlea, the vestibulocochlear nerve. There's the auditory tube. Close-up of the internal ear. We see the widened areas or ampulla of the semicircular canals. There is the oval window underneath the stapes. There's the round window. And then when we look at the cochlea, we can see scala tympani, cochlear duct, and scala vestibuli. So, the internal ear's cochlea. Let's talk about that in more detail. Again, it is a membrane structure filled with fluid found within a bony structure that's shaped kind of like a snail shell that is also filled with fluid. So there end up being 3 fluid-filled chambers. You have the scala vestibuli, which is the superior chamber. You have the cochlear duct, which is the middle chamber filled with endolymph, and then inferior to that is the scala tympani, the inferior duct. And so the scala vestibuli starts at the oval window, receives the vibrations from the oval window, and can carry it up along the cochlea. The cochlear duct is where the vibrations will eventually pass through, and then scala tympani ends in the round window that then allows excess vibrations to dissipate. So again, it is the vibrating stapes within the oval window that is going to generate fluid pressure waves that travel along the scala vestibuli and up the cochlea. And then the excess pressure waves at the end get released via the round window. So within the cochlear duct, we have the receptors for hearing in a structure called the spiral organ. It's spiral, S-P-I-R-A-L. And it is called that because, again, it is coiling up the spiral of the cochlea. And it is able to detect the fluid pressure waves as they pass through the cochlear duct and stimulate the proper receptors. So here is a close-up of the spiral organ. We see that it has a tectoral membrane, a cartilaginous structure. Then underneath that, you find a cellular structure with many epithelial cells that help to hold in place the hair cells being the receptors. So there are outer hair cells, inner hair cells. They all have these little hair bundles that protrude upward and are embedded in the tectoral membrane. And so when pressure waves come down and hit the tectoral membrane, it causes it to vibrate. This bends the hairs of the hair cells, and by bending the hair bundles, that is the signal. That is the signal that gets transmitted to the sensory neuron. So the way this all works is we have sound waves. These are air pressure waves traveling down the external auditory canal. They collide with the tympanic membrane, causing it to vibrate. The vibration of the tympanic membrane then transmits to the malleus, which then transmits to the incus, which then transmits to the stapes. And the stapes then fits into the oval window. And the point of these bony structures is to increase the intensity of that vibration by something around 20-fold. Then the vibration in the oval window, the bulging in and out, will lead to fluid pressure waves in the perilymph of the scala vestibuli, and those pressure waves will begin traveling up the cochlea. In certain areas, depending on the frequency of those pressure waves, they will pass through the wall into the cochlear duct, into the endolymph. There they will hit the tectoral membrane of the spiral organ, bending the hairs of the hair cells, generating the nerve impulse. And then the excess vibrations will be released out the round window. Now, the cool thing about the cochlear duct is it is tuned, as it were, so that different regions of it will allow the vibrations of a specific pitch, so that it's kind of like a piano keyboard where certain keys produce certain pitches. Here, certain areas allow for certain pitches to pass through. All right, so we have that first order sensory neurons. They are the ones that synapse with the hair cells. So when hair cells stimulated, the nerve impulse will travel through the first order sensory neurons that are traveling through the vestibulocochlear nerve. They will then synapse with the second order sensory neurons within the medulla oblongata, and they will get transmitted up to the thalamus, where the second order sensory neurons will synapse with the third order sensory neurons that will then carry the signal to the primary auditory area in the temporal lobe. And what you can see here is that a lot of that signal will go to the primary auditory area on the opposite side of the ear, which is pretty wild. All right, some disorders associated with hearing. We have otitis media. Otitis media is the acute infection of the middle ear, and happens a lot, especially in children, where bacteria are able to pass up the auditory tube and infect the middle ear. Then there's, of course, deafness. There's sensory neural deafness. Sensory neural deafness is caused by damage to the cochlear branch of the vestibulocochlear nerve. So the nerve itself is damaged, preventing the transmission of that signal. And then there's conduction deafness. Conduction deafness is when there's some kind of impairment or damage in the external or middle ear. So it is a structural damage to the mechanisms for transmitting the vibration versus a neural damage to the nerve itself. Now there's this device called the cochlear implant, which is able to bypass conduction deafness. So it's able to send a signal directly to the nerves in the ear, nerves within the spiral organ, so that the person can hear again. But again, this only works if it's conduction deafness, if it's damage to external or middle ear mechanisms. You must have a functional sensory nerve for it to work. All right, that is it for this part.